Hello students, I am Sabum Courtney, a student doctor at the Faculty of Medicine and Biomedical Sciences from the University of Yaoundé 1. Today we are going to be having a general overview of what embryology is all about. Let's dive directly to our objectives. Now at the end of these lessons, the students are expected to be able to differentiate between the embryonic and the fetal period. Describe the steps involved in embryogenesis. Know some teratogenic drugs. Now what is our plan? We have introductions, some definitions, stages of embryogenesis, fetal annexes, teratogens, prenatal diagnosis, some abnormalities, and conclusion. Now, embryology is the fundamental science of medicine. It helps us to understand the underlying mechanisms of human development. Why is embryology so important in medicine? Embryology is important in medicine because with an understanding of the mechanisms underlying human development, we are able to pinpoint the errors that are found in congenital diseases and be able to know the essential ways to prevent these diseases from taking place. Now, if we must understand embryology, there are some basic definitions we must know. Embryogenesis. Embryogenesis is the formation of the embryo. Now, embryology is a study of the developmental events or the series of events occurring during prenatal period, that is the period before birth. Now the prenatal period is divided into subgroups based on the certain events that occur within this specific periods. That is, these sub-periods have specific events that occur within them. Now, what are these periods? The first is the pre-embryonic period, which begins from the first to the second week of development. The events occurring within this period is the formation of the zygote, and the zygote, which is actually unicellular, becoming a multicellular structure. The embryonic period. The embryonic period begins from the 3rd to the 8th week of development. Now, during this period of time, the most important event occurring at this level is organogenesis, which involves the formation of our organs. The next is the fetal period, which begins from the 9th week and runs up to the time of birth. At this point in time, the events occurring here are just organ growth, organ maturation, and the organs gain functionality. Pregnancy, or the prenatal stage. Pregnancy lasts for 37 to 42 weeks. Now, embryogenesis begins with the formation of the zygote. Now, if we must understand how the zygote comes into being, we must understand understand how the gametes that fuse to form the zygote come into place. That is why gametogenesis is so important. Now, gametogenesis is the process by which gametes are being formed. Now, we have oogenesis, which occurs in females, and spermatogenesis, which occurs in males. Now, oogenesis is the formation of the egg cell from the parent cell, that is the oogonia. Now, the oogonia is a parent diploid cell which is found in the ovaries. Now, the oogonia begins its journey, its differentiation towards the ovum by beginning first with a series of mitotic division to form the primary oocyte. These processes occur before birth. Now, the primary oocyte undergoes its first meiotic division still before birth, but it is arrested at prophase 1. Now, at puberty, the primary oocyte, under the influence of gonadotropins, completes the first meiotic division. But unlike the normal first meiotic division found in other cells, that of the oocyte is actually different, where there is an unequal sharing of the cytoplasm, where one of the cells takes majority of the cytoplasm to form the secondary oocyte, and the other cell takes less or little cytoplasm to form the first polar body, which will later die in time. Now, the fate of the secondary oocyte, will depend if fertilization occurs or not. That is, if the secondary oocyte is fertilized or not. Now, if the secondary oocyte is not fertilized, the oocyte lasts for maximum of 24 hours after ovulation. But if the secondary oocyte is fertilized, it completes its second meiotic division. Now, the secondary oocyte, after the completion of the first meiotic division, to form the secondary oocyte, the secondary oocyte begins the secondary meiotic division, but it is arrested at metaphase 2. Now, if the egg is fertilized, if the secondary oocyte is fertilized, it completes its second meiotic division with the formation of two daughter cells, with one of the cells also carrying majority of the cytoplasm to form the ovum, and the other cell carrying less cytoplasm to form the second polar body, which will also eventually die with time. Now, this diagram gives us an understanding of what oogenesis is all about. Now, during the embryonic period, we see that the parent cell, that is the ogonium, is diploid, that is 23 pairs of chromosome. Now, it undergoes mitosis to form the primary oocyte, which undergoes the first meiotic division, but it's halted at prophase 1. Now, it remains arrested until puberty, where under the influence of gonadotropins, what happens? It completes the first meiotic division to form the secondary oocyte and the first polar body. You see, the difference in size explains the unequal sharing of the cytoplasm. Now, the secondary oocyte begins the second meiotic division, but it is halted at metaphase 2. Now, the completion of the second meiotic division will depend on if fertilization occurs. That is, if the sperm fuses with the egg. And this also explains like follicular development, the development living from the primordial follicle to the mature Graafian follicle, or the vesicular follicle. Now, spermatogenesis. Spermatogenesis is the formation of sperm cells from the spermatogonia. That is the formation of a haploid sperm cell from a diploid spermatogonia. Now, the spermatogonia resides in the testes, as the oogonia resides in the ovaries. Now, the spermatogonia firstly undergoes a series of mitotic divisions to form the primary spermatocyte. Now, the primary spermatocyte the primary spermatocyte, as we see from here, the spermatogonium first of all undergoes a mitotic division to form the primary spermatocyte. Now, the primary spermatocyte begins the first meiotic division to form the secondary spermatocyte. Now, the secondary spermatocyte undergoes a second meiotic division to form what? To form 4 haploid spermatids. Now, these spermatids are not, I repeat, these spermatids are not mature sperm cells. Now, in order for the spermatid to form mature sperm cells, or the spermatozoa, it must undergo a process known as spermiogenesis. Where what happens? Spermiogenesis, which is the process, it is the process by which the spermatids undergo morphological changes to form mature sperm cells, or the spermatozoa. What are these morphological changes? The first is the shedding of the cytoplasm. The cytoplasm is being shed. The second is the formation of the head, the tail, and the neck. Now, the third important process is the formation of the acrosome, which is very important for the hydrolysis of the zona pellucida. Now, all of this explains what we have just said. Now, we are going to move straight to embryogenesis. Okay, with a brief summary of what gametogenesis is all about, we are going to dive directly to embryogenesis. Embryogenesis actually begins with the formation of the zygote during fertilization. Now, embryogenesis is divided into periods. That is the pre-embryonic period, the embryonic period, and the fetal period, in which in each of these periods we have certain and specific events occurring within these periods, as we are going to see. Now, as we saw from the very beginning, the pre-embryonic period begins from fertilization up to the 2nd week, and the embryonic period begins from the 3rd week to the 8th week of gestation. The most important aspect it's been recognized, the most important event that is being recognized during the embryonic period is organogenesis, that is the formation of organs. Now, this process, this period is a very fragile phase. It is a very fragile period because the result of the work done at this level is maintained and it is permanent throughout the process of development. That is, in case of any error, the error is kept permanent. The error is maintained throughout the process of human development. Now, the first week begins with fertilization, that is the formation of the zygote, where fertilization is just the fusion of the haploid secondary oocyte with the haploid sperm cell to form what? A diploid zygote. This occurs at the external 1/3 of the fallopian tube at its specific position known as the ampulla. Now, for the sperm to fertilize the secondary oocyte, it must undergo a process known as capacitation, which is a process that occurs for 7 hours, where there is the shedding of or the removing of the acrosomal membrane. And it must be clearly noted that only a capacitated sperm can cross through the corona radiata. Now for the sperm cell to arrive and fuse with the secondary oocyte plasma membrane, it must hydrolyze the zona pellucida, the glycoprotein layer which surrounds the plasma membrane of the secondary oocyte. It does this it hydrolyzes this zona pellucida by releasing hydrolytic enzymes from the acrosome in a reaction known as acrosomal reaction. Now, the fusion of the egg cell and the sperm cell, of the egg cell and the sperm cell creates a type of reaction known as the cortical reaction, which prevents further entry of another sperm, to prevent a situation known as polyspermy. That is when more than one sperm cell fertilizes the secondary oocyte. Now, if you look at the diagram explains everything. Here at the level of the head, here we have our acrosome. Now we see that this is our secondary oocyte, being surrounded first by the outermost layer, that is the corona radiata, made up of granulosa cells. Now we have also a glycoprotein layer known as the zona pellucida.
It's not good, eh?
And also we have, now when the sperm hydrolyzes the zona pellucida, they release into the cytoplasm of the oocyte their pronucleus, which actually fuses with the pronucleus of the ovum. Now, after the formation of the zygote begins to undergo rapid mitotic division. That is a rapid series of mitotic divisions in a process known as cleavage. Now, it goes through a process of 2-cell stage, a 4-cell stage, 8-cell stage, 16-cell stage. Now, when it contains 16 cells, the zygote is known as the morula, and the morula is found at day 3. Now, when it continues from the 16-cell stage to the 32 to the 64-cell stage, these cells reorganize themselves into an inner mass known as the embryoblast and an outer mass known as the trophoblast, with a fluid-filled cavity known as the blastocoel. At this point, the zygote is now called the blastocyst. Now, implantation is the attachment of the zygote to the endometrial walls of the uterus, specifically the superior to third portion of the endometrial walls of the uterus. The diagram, as you can see, the diagram explains it all. The formation of the zygote, as you can see, which is unicellular, moving through cleavage to form a multicellular structure, as you can see, with the formation of the morula, that is a 16-cell stage, and the formation of the blastocyst, where these cells reorganize themselves into an outer cell mass known as the trophoblast and an inner cell mass known as the embryoblast, with a cavity known as the blastocoel. So this entire structure is now what we call the blastocyst. Now, the second week of development is actually the formation of the bilaminar layer. That is, the embryo at this stage has a bilaminar disc. When we mean the bilaminar disc, what happens? The embryoblast forms a 2-cell layer, that is the epiblast and the hypoblast. The trophoblast also divides into an inner cytotrophoblast and an outer syncytotrophoblast, which secretes the human chronic gonadotropin, which is the hormone of pregnancy. That is, it is used to detect pregnancy. Now, the third week now is the formation of a three germ layer. That is the process known as gastrulation. Now, in the third week, the event occurring here is the formation of the 3-germ layer in a process known as gastrulation. Now, what are these 3 germ layers? You have the ectoderm, the mesoderm, and the endoderm. And it must be clearly noted that all the three germ layers are formed from the epiblast only and not from the hypoblast. Now, each of these germ layers have their derivatives. Each of these germ layers have what they are going to differentiate into, their specific structures that they are going to differentiate into. Now let us have a brief overview of the derivatives of the three germ layers. We will begin with the ectoderm. The ectoderm, the derivatives include, you have the epidermis, the nervous tissues, sense organs, pituitary gland, adrenal gland, the enamels, the lens of the eye. The mesoderm involves the dermis of the skin, muscular tissues, connective tissues, the adrenal cortex, the dentine of the teeth, the heart, the kidneys, the ureters, the internal reproductive organs, the spleen. Now, the endoderm. The endoderm involves the derivatives here include the liver, the guts, the pancreas, the thymus, the tonsils, the parathyroid glands, and the thyroid glands. Now, these diagrams gives us a clear overview of how implantation actually takes place. As you can see, this is the blastocyst. The blastocyst actually attaches to the endometrial walls of the uterus using what? Using the outer part, the outer layer of the trophoblast, that is the syncytotrophoblast. And at this point in time, we notice that what between the cells of the epiblast, there's a development of a fluid filled cavity, that is the amniotic cavity, and the hypoblast cells form another cavity, which we call what? The yolk sac. Now, at the level of week 4, we have two specific foldings, that is the cephalocardial folding and the lateral folding. The cephalocardial folding helps to form the head and the buttocks, while the transverse folding helps to form the trunk. Now you see it's clearly seen that from the 4th week to the 8th week is organogenesis. Now this diagram also gives us the clear explanation. This is the formation, this is around this cephalocardial folding, that is the head and the buttocks. Now this is a transverse folding which will give rise to the trunk. Now the filter period. Now the filter period is the events that actually occur at the level of the filter period are organ maturation and organ growth, and it begins from week 9 to birth. Now the filter annexes. The filter annexes are structures or organs that are necessary for the survival of the fetus. Now, what are those organs that are necessary for the survival of the fetus? They include the placenta, the umbilical cord, the chorion, the amnions, the yolk sac, and the atlantius. Now we are going to begin with the first, the placenta. The placenta is fully formed at the 12th week of gestation. Now it is an organ that permits exchange between the mother and the fetus. The placenta is actually made up of two parts. That is the maternal portion or the mother's portion, that is the deciduum basalis, and the fetal portion or the fetal portion, which that is the chorion frondosum. It has a weight of about 600 grams and a length of 15 to 20 centimeters with thickness of 2.5 centimeters. Now, what is the function of the placenta? The placenta has a nutritive role, that is the exchange of glucose, amino acids, fatty acids, minerals, vitamins needed for the growth of the fetus that are gotten from the maternal circulation. Now, excretion. Normally, its metabolic activities leads to the production of ammonia, urea, uric acid, creatinine that will be very toxic if they accumulate within the fetus. So they must be excreted since the urinary system of the fetus is not yet functional. So they depend on the mother's circulation for excretion. Respiration. Now, because the lungs are not yet fully functional, they totally depend on the mother's blood for oxygen supply and the removal of carbon dioxide. Endocrine role. Now, the placenta is involved in the secretion of estrogen, progesterone, and HCG. Immunity. Now, the placenta, although has a barrier, although the placenta forms the placenta barrier, the antibody immunoglobulin G is able to cross through the placenta barrier, and it is involved in immunity, that is fighting against foreign bodies within the fetus. The umbilical cord. Now, the placenta itself is the connection between the fetus and the mother's circulation. Now, what is the connection between the placenta and the fetus? It is the umbilical cord. The umbilical cord connects the placenta and the fetus. Now, it is a rope-like structure that is actually tough, with length from 30 to 100 centimeters long. It is covered by a whitish connective tissue known as the Watson's jelly. Now, it contains 2 umbilical arteries and 1 umbilical vein. Now, unlike our normal circulation and its blood vessels, the 2 umbilical arteries carry deoxygenated blood, while the umbilical vein carries oxygenated blood. Now we continue directly to the amniotic membrane. Now it is made up of, you have the amnios, which makes up the internal membrane that is in direct contact with the amniotic fluid. Now we also have the chorion, which makes up the external membrane that surrounds the amnios, together with the yolk sac and the umbilical cord. Now between these 2 membranes, you have the amniotic fluid, which we know that is the fluid that actually bathes the baby and has some functions like preventing, like a shock absorber. Now the yolk sac. The yolk sac is a membranous sac attached to the embryo and formed from the hypoblast. That is the cells of the hypoblast. Now it has basically 2 functions, like involved in the first site of cells of erythropoiesis, together with also cells of the gonads. We have the last, which is the atlantios, which is a sac-like structure. Now, teratogens. Teratogens are substances that cause a disruption or malformation during the process of fetal development. Now, some of these chemicals that actually cause this disruption include, you have herbicides, the drugs, you have tetracycline, doxycycline, radiations like gamma, tobacco, alcohol, and some infections, like this is an abbreviation, like you have Toxoplasma gondis, rubella, cytomegaloblastic viruses. Prenatal diagnosis. Now, some of these prenatal diagnoses include, we have amniocentesis, That is the taking of samples of the amniotic fluid and analyzing them, which is done at the 14th to the 18th week of gestation. We have chronic villi sampling, which is a genetic analysis of the chronic villi and can be done as early as the 8th week of gestation. It shows the risk of abortion. You have ultrasound, which shows the morphology of the fetus. Now, some pathologies during prenatal development include neural tube defect. That is, the neural tube fails to close completely, and this is usually as a result of lack of folic acid, that is vitamin B9. Now, some consequences of failure for the neural tube to close includes spina bifida and anencephaly, that is the absence of the brain. We also have ectopic pregnancy, that is the zygote fails to implant on the uterus. That is, if it implants outside of the uterus. You have, for example, other sites of implantation. You have the fallopian tube, you have the ovaries, you have the abdominal cavity itself. So we have finally come to an end of this lesson. And it must be clearly seen that the mechanisms underlying embryogenesis really gives us an understanding of how human development takes place, and really helps us to identify or pinpoint the errors seen during congenital diseases and how we can really prevent them. Thank you for your kind attention. Are there any questions?